It's my pleasure to introduce today Dr. Jennifer Elisseeff. She got her BS in chemistry from Carnegie Mellon, and then got her PhD in medical engineering at MIT and Harvard in the Joint Health Sciences program, studying with the famed Robert Langer. Among her many accomplishments, she started two very successful companies, and is a member of the National Academy of Engineering and of Science. So looking very forward to hearing her talk about the immune system, regeneration, and fibrosis today.
Thank you very much. I'm really excited to be here today, and really excited talking to people about aging and metabolism and all of the really interesting research going on here. So I actually come from the field of tissue engineering. So I started out looking in biomaterials and making different polymers that could be built into three-dimensional scaffolds to hold cells, and along with biological signals. So this field evolved about, I guess, the late 80s as a method to rebuild and regenerate new tissues. So biomaterial scaffold development, where we could put in biological cues, and in the late 90s, the discovery of adult and embryonic stem cells was where we put a lot of our early efforts in designing biomaterial scaffolds to modulate stem cell function and build new tissues. So obviously the clinical need is important. Organ transplant is growing, but it still has many issues with immunosuppression that's required. For other places where you get tissue loss, such as osteoarthritis, where you lose cartilage, the endpoint is a metal or plastic implant. So the idea of rebuilding your native tissue is of great interest. And you'll see at the end, and my particular interest connecting here is through aging, which is also a place where we lose tissue and organ function. Clinical translation was slow in tissue engineering. So when I started about 20 years ago at Hopkins, we were interested in looking at stem cells and building materials for that, but we were also interested in what can we do to help patients today, and what can we take so far of what we've learned and develop with clinical therapy. So clinical translation is great because you get to hopefully improve patient care, but it also helps redirect research. And number one, you can start looking at maybe different therapeutic targets. So it led us to maybe think that stem cells were not the place to go. So our first clinical trial in cartilage repair showed us that, well, we weren't following that paradigm of the biomaterial scaffold serving as a 3D framework. We were actually redirecting the repair process and modulating scar formation and reducing that scar formation to allow for more repair. And then we had a second trial, which I joke was our dark side project because it was involved with wrinkle fillers, but we got some really cool data of biomaterial responses in people. And so these trials really led us to step back and say, hey, maybe we need to look at the immune system, and maybe this is a more important regulator of tissue repair than stem cells, or maybe it's a little bit upstream. So we wanted to step back, understand the biomaterial immune response, and really target that for repair. So I like to term this our lessons in translation. So a pretty big shift from biomaterials development and stem cell research to all of a sudden immunology. The concept of regenerative immunotherapies for tissue repair is where we're going. So the concept of regenerative immunology really marries together regenerative medicine, immunology, and tissue engineering. And the first thing that we've been doing is mapping the immune response to different types of injuries and materials. So people have looked at innate immune responses, and I'll go into some immune system terminology in a minute. But now many people have looked at the adaptive immune system, in particular T cells. So I'll mostly be talking today about T cells. Well, this immune system background really sets the framework for downstream tissue repair processes, such as mobilization of stem cells and things like senescence. And can we use this information to engineer immunotherapies to create a pro-regenerative environment? And so we're taking the approach of regenerative immunotherapies. In part, there are connections with cancer immunologists who remind me frequently that the immune system is therapeutically accessible. There are a lot of great drugs to target the immune system. So this could be an interesting target for regenerative medicine. Now, as a newcomer 10 years ago to the immune system, I recognized that the nomenclature and the whole language of immunology can be challenging. So please feel free to stop me anywhere if I need to review some. But here's some basics. So we have the innate immune system, which tends to respond faster to stimuli, but with, let's say, less specificity or memory. So macrophages have been studied for a while in the context of regeneration. Neutrophils, a lot of work there. But as I mentioned earlier, less so in the T cell area. So T cells can either be CD4 positive or CD8 positive, and the CD4 are helper T cells, and they help by making cytokines, and then there are different classes of those helper T cells. So we're going to be talking just about two of those classes of helper T cells. I'd be happy to talk later as we're starting to explore CD8s and regulatory T cells, but we'll focus today just on two types of T cells. These cells are really interesting because of their specificity and memory and really their long lifetime. So a naive T cell connects with an antigen-presenting cell, and there are three signals that need to get activated, and we'll talk about those details in a little while. But there are all these different pathways that T cell can go down depending on the cytokines and the environment in which it's found. And what I find interesting about the immune system is that it's historically been studied in the context of host defense and infections. And we're really seeing new functions of the immune system as we're looking at injury and aging. So this Th1 phenotype, interferon gamma, this is what cancer people like, right? Because you're going to help kill cells. And it's involved with viral responses to viral infections. And then we have this Th2 group here, which is connected with helminth infections, allergies, but we're seeing that it's quite important in healing. Th17 is connected with mucosal bacteria, things like tuberculosis, but it's also been found to be important in autoimmunity, and I'll be talking about in the context of fibrosis. And an interesting note that we'll be talking about again is that this pathway actually inhibits that one, right? So you can either go one direction or the other. And then, unfortunately, I won't talk too much about Tregs today, but that's an area of active work in the lab now. So you can have all those different cell types, but then you can also have categories of immune responses. So type 2 immune responses are characterized by production of a number of different cytokines. These are interleukins. Interleukin 4, 5, and 13 are connected with this type 2 immune response. And as I mentioned, it's connected with host defense and these various both good and bad things. Now, what's important to remember with the immune system is that you've got a lot of cells working together. So a number of cell types can be making this type 2 immune signature, including eosinophils, innate lymphocytes, and these Th2 cells, which are the helper T cells. And they're presenting antigen on the T cell receptor, and that's recognized by recognizing different peptides in the antigen presenting cells. So we're just starting to look at sequencing these and understanding those interactions. Don't worry, we won't get into that. So the type 3 or IL-17 type 17 response is characterized easy enough by production of IL-17. Along with some other factors, IL-21 and IL-22, and again, host defense, and actually no real good things. Actually, the good things it's connected with are vascularization and wound healing, but it's also connected with autoimmunity and fibrosis, and I'll show you today aging. So which cells can make these type 17 cytokines or signature of immune response? The one interesting thing here is gamma delta T cells, which I'll be talking about. Gamma delta T cells are, let's say, they're not too new, but a newer cell type. And they have a different receptor than your Th helper cell that has an alpha beta TCR. This one has a gamma delta TCR. And it doesn't have the same ligand recognition. And frankly, people are still trying to understand what gamma deltas are responding to. And then innate lymphocytes can participate in any of them. So those are the primary immune responses we'll be talking to, and I'll go over, again, any nomenclatures as needed. So I started off with biomaterials, and as we looked at biomaterials that promoted regeneration through, let's say, known immunological mechanisms, these biological scaffolds were recognized as being modulatory for macrophages and pro-regenerative. And essentially what these biological scaffolds are taking different extracellular matrices from tissues, either porcine or human. So products on the market are derived from urinary bladder matrix, small intestinal submucosa, demineralized bone, a number of choices. And you essentially have an ECM matrix. A particular note, as we've studied those matrices, you also have cell remnants, because when you process these ECM or these tissues, you break up and kill the cells, but the remnants of the intracellular proteins are sort of stuck on the extracellular matrix. And that's important because they can be signals to the immune system. Anyway, they're used clinically for a number of applications. Our first sort of in-depth immune project was with Caitlin, looking at applying these scaffolds to a muscle wound. So I would categorize a muscle wound in general as a wound that heals pretty well, at least when it's small enough, because you're exercising all the time, and hopefully, as you're walking around or actual exercise, and that damage to the muscle is pretty good at repairing. But if we put a scaffold in there, let's say with a larger defect, and just look at the T cells and what kind of T cells we see, it depends on the time point, but essentially what these biological scaffolds do is they give a power boost to those helper T cells, the CD4, making IL-4, that's just one marker cytokine that we can use, and that's the Th2 phenotype. So you have a few of them in just a control wound, but here with that biological scaffold, you increase it. Why is that useful? Well, that actually modulates the macrophage behavior. So macrophages shift their polarization to a more regenerative phenotype. And Caitlin was a very astute PhD student, and she recognized that the draining lymph nodes looked different when the biomaterial was in place and looked at what they were making. And both the local draining lymph nodes and distal lymph nodes had increased expression of interleukin-4, representative of this Th2 phenotype. And I was very fascinated about this because essentially we think of a wound as being local, but it's really not. You can have systemic effects from that local wound. And that also introduced the concept that systemic factors can influence the local wound response. So this was our first foray into the T cell response to wounds and materials, and being able to modulate that. How do you take that to actually therapeutic development? We looked at nature for inspiration here. So this is a parasite helminth, which you might recognize was in one of the functions of the type 2 immune response. And these parasitic helminths are actually recognized for making a type 2 immune response. And we can sort of modify that for our needs to promote regeneration. So we can take these eggs that come from the schistosomiasis, for example. We're now looking at H. polygyrus. We can break up the eggs and essentially purify various components, a lot of screening here, but ultimately to create what we call a regenerative soluble egg antigen, or SEA formulation, to stimulate type 2 without the negative effects of an infection of the worm. So this is looking at some type 2 cytokines, interleukin-4 and 13, that go up with the regenerative SEA. And this one that we had to sort of purify to get a reduction in the inflammatory components that we want to avoid. And just one injection at the time of injury, in our draining lymph node, we see that IL-4 change on the order of weeks. That's a nice thing about T cells. They live for a while, and you can get a long-term effect by modulating T cells. I'm going to be talking about IL-17 and from gamma deltas and T cells later, so I won't get into that right here, but I do want to show you that this is a stain looking for fibrosis. And so when you have a really big injury, you get this fibrosis, and when we have the regenerative SEA, we can reduce that fibrosis. And I did forget to put my only metabolism slide into this. So what we see with the ECM material is if we put in T cells in a mouse deficient of T cells and B cells that has a deficiency in the TORC2 complex, the cells can't go under Th2 differentiation, and we get more fibrosis. So when you don't have that Th2, you get more fibrosis and more adipogenesis. We can encapsulate these formulations into biomaterials and get a more extended release of that SEA, and we can get better tissue repair there. So that gives an idea of a regenerative response, but we're also interested in what happens in the more fibrotic response, where if you have a dysfunction in your mTOR complex where you can't undergo Th2 differentiation, or you're somehow having it blocked, what happens? So biomaterials are nice models for things like fibrosis. And there's been a study of this response since the 1970s. So what happens when you put a synthetic material in the body is that you have proteins sticking to the surface, depending on the surface chemistry. Neutrophils first come, and then you get monocytes recruited that will differentiate into macrophages, form foreign body giant cells, and you get activation of fibroblasts. Now, when I spoke a number of years in Seattle, there was someone from the group who did some of the first studies in the 70s, where they had this cage implant system to look at the immune cells responding to implants. And whenever they saw lymphocytes or T cells in the response, they threw it away because they said, oh, that must be a contamination. It doesn't matter. But what's nice is all these new technologies, really pioneered in part by the cancer immunology field, we can use here and understand a little bit better that response. Now, this is a sample from people. There's some interesting things I want to point out. So this is the fibrotic area, right? So you get that thick fibrosis, and then you have these large blood vessels that are next to that area of fibrosis. So we think these blood vessels have trouble penetrating into that fibrosis, and those blood vessels are where you have immune cells coming in and infiltrating the region. So what's nice about this, so we can get samples from the clinics. So when people are having, for example, post-mastectomy reconstruction, a temporary spacer is implanted for a few months before a permanent implant is placed, and we can get those back. So it's only been in there a few months, and we also get implants that have been for a while and essentially failed because of that fibrosis. What we found is, well, these T cells are not making more interleukin-4 like the materials I showed you before, but more IL-17. And we see that IL-17 coming from the helper T cells and from gamma delta T cells. So we have the human data, and then we go back to the clinic. We go back to, from the clinic to the lab with some mouse models, and we can implant some polymers that we know induce a robust fibrosis and immune response. So this is a picosero staining where you can see the collagen organization and the amount of collagen. And if we give an anti-IL-17 ANF antibody, we can reduce that fibrosis around the implant. Now, I told you I would tell you more about that T cell response. And here it is. So you have this T cell receptor here that will recognize peptides presented on this MHC complex. And then you've got various co-activation signals. So you have a signal 1, signal 2 with this another activation, and then cytokines can provide your signal 3. So what in the world is actually being recognized on this MHC when you have a plastic material, essentially a synthetic polymer? It's not a peptide. So to determine if it was antigen specific, we did this chimera study. So we took wild type donor cells that have CD45.1, so we can tell it apart from an OT2 RAG CD45.2. So this RAG knockout animal has helper T cells that only recognize ovalbumin. That's the only TCR receptor recognition that they have. And we know that there's no ovalbumin in our system. So if they are activated, it's non-specific, meaning it's not going through that TCR complex. So we can make sure that there's a response from both of those T cells and that mouse that's a chimera. We essentially only get a response of IL-17 when we have the wild type cells. So those OT2 knockout T cells don't get activated. So it's an antigen specific response. And we have some ideas about what the antigen is, and that's where we're doing some TCR analysis. I showed this slide before, and I didn't mention this part. So it's well recognized that IL-6, IL-1 beta, TGF beta, and these other factors are involved in that differentiation towards this fibrosis phenotype, which blocks that regenerative phenotype. IL-6 and IL-1 beta are made in part by things like senescent cells. So those fibroblasts are the ones actually making that extracellular matrix. And I promise I'm going to tie this to aging soon. So fibroblasts were thought, say 25 years ago, I was told they're sort of like the cockroaches in the body. When there's an injury, they go real quick and seal it up, but they're not very smart. They don't make very good tissue, right? Scar tissue is weak and knows. Well, single cell technologies now have defined the heterogeneity and immunological activity of fibroblasts. So they're not so simple, and they play a lot, they play a big role in tissue homeostasis and immune connections. So in arthritis, people have shown subtypes of fibroblasts, cancer-associated fibroblasts, the myofibroblasts and inflammatory. And then Shannon Turley's done some beautiful work on different healthy, undifferentiated versus pathological fibroblasts. So one place that we've been looking at fibroblasts is the concept of cellular senescence. So this can be defined as proliferative senescence, where cells that you might be culturing in a dish stop proliferating, essentially get tired, reduce telomeres, and just reduce the ability to proliferate. But there's also many different ways to induce senescence with oxidative damage, stress, oncogenic stress, and I'll be talking more about inflammatory induced senescence. So senescence is characterized by a lack of proliferation, so the cells are not dividing, but they're far from quiescent. They're busy secreting a senescence-associated secretory phenotype, or SASP, and the SASP is implicated in their biological function or their association with things like tumor promotion, a number of diseases associated with aging, including arthritis, heart disease, diabetes, neurodegeneration. However, they're also associated with good things like tissue repair and even embryological development. And these SASP factors include cytokines such as the IL-6, IL-1 beta, and things that will help modulate those T cells to a certain phenotype. So P16 is, albeit imperfect, a way that people look for cellular senescence. And if you just look here at the staining for P16, we see it around that fibrotic area when we put in those materials. So we have these senescent cells that develop. Now, what's their connection then with the immune response? Well, here's P16 in a wild type mouse. When we take out IL-17 or IL-17 receptor signaling, we have reduced senescence. And in the breast capsules and fibrosis from the clinic, we see a correlation between P16 and IL-17, and we can see plenty of P16 positive cells in that fibrotic area. So we can culture together just in a dish to see what's going on here. A senescent fibroblast, which is artificially induced, so not perfect. It's artificially induced, and if we culture it next to a naive T cell, we can induce IL-17 differentiation. And that's in the presence of TGF-beta. If TGF-beta is not present, it's interferon gamma. It's very sort of clear switch. We can go the other way around. We can induce naive T cells towards the Th17 pathway with various cocktails, culture them next to healthy fibroblasts, and induce senescence in those healthy fibroblasts. So there's this feed-forward loop between that fibrotic Th17 signature and senescence. And this is what we look at as an inflammatory induced senescence, which we think is more physiologically relevant. So when we think about therapeutic targets, you need to hit both pathways. If you hit just senescence, you're going to have more senescence develop because you haven't targeted that Th17 or IL-17 production. So you need a combination therapy. So inflammatory markers are up here. When you have a combination senolytic plus the neutralizing antibody for IL-17, reduce P16, IL-17, IL-6, and other cytokines, and then reduce TGF-beta and your matrix production. So I've given you sort of two extremes of a very regenerative Th2 response and a profibrotic Th17 and senescence signature. But we haven't we've scratched the surface on what's really going on, and we want to characterize more who's involved. And so we use a combination of advanced flow cytometry, some single cell atlases that we're building, and then bulk sequencing of various specific subtypes. And with that, we've been building a big repertoire of data sets for the regenerative and fibrotic materials, and also developing programs that can help predict cell signaling. So there are a number of programs out there to look at receptor ligand expression. We go a little bit backwards, that we start with transcription factor targets and looking at their activation within our data sets. And then we can predict transcription factor activation, and then we look for expressed receptors and ligands. So just because you have a receptor and ligand being expressed doesn't mean you're going to actually have it signaling in a particular cell. So I think it is important to look at that transcription factor activation. And so from this, we're going to try to tease out what type of immune stromal communication is happening with the idea of tissue homeostasis. You've got your resident immune cells, and you've got your vascularity, and you've got your matrix and your tissue-specific cells, or whatever cell type. When there's some disruption, whether it's a tumor or an injury, or I'd argue even aging, you get disruption of the vasculature, your matrix, cells within the matrix are activated, and you've got immune cells infiltrating. And how these cells communicate will determine those outcomes of regeneration versus fibrosis. So we're particularly interested in using our models as engineers to define senescent cells or other smaller subpopulations, because it's a model where we can enrich a target, and we can do a better analysis when it's enriched. And then we can apply it to more environments where it might be a little bit more fuzzy or gray. So fibrosis, as I showed, is a model for senescence enrichment. And senescent cells are, as I mentioned, implicated in multiple diseases like lung fibrosis, atherosclerosis. We've seen it in fibroids, and it's also connected with aging. So there is a generalized fibrosis with aging that occurs, and there are conserved aspects across tissues, and of course, tissue specific. So we first started using a reporter model, and this technique can be applied for any small population of interest. We used a reporter for P16, developed by Jan van Derzen at Mayo. And once P16 is expressed, it's turned on. And we can apply the model that I just showed you, putting in those polymers and synthetic materials, and we can get some nice fibrosis here and nice senescence. And using flow cytometry, we can see which cells are senescent. And what's interesting here is I want you to see how few cells are senescent. So, be less than 1% of your population of cells is senescent. And we can see which cell types are senescent. So primarily in a fibrosis in a young animal, it's CD29 positive, CD45 negative. So it's a stromal cell. So then we can actually pull out those cells because the reporter is strong enough and we've enriched enough. It's still not easy. There's not many cells per animal. But we get a small population, and we can do bulk in-depth sequencing on those cells sorted from that model environment. And with that signature, we can start looking for gene pathways, and it was a little bit unexpected. 2 pathways. So things related to vasculature development, endothelial migration, vascular development, and then chondrocyte differentiation, which, sure, that's great. We started out with cartilage, and we've been looking at arthritis. But is this just a contamination? What's going on here? It actually wasn't a contamination, and there's a subpopulation of senescent cells that make the fibrotic tissue, a lot of matrix, and they have matrix molecules that were thought of as being really specific to cartilage, but they're actually in fibrosis and scar. Then working with computational colleagues, they have a program called Project R, where we can take that signature of that senescent cell that we've sorted out, very small population, and then apply that across target data sets to see if we can estimate it. So this is an example. This is a single cell data sets, and we have a projection of cells that are similar to that senescent phenotype in our model that you wouldn't normally be able to capture with single cell. And the red cells are the ones that have high similarity. And there's 3 populations pretty much. There's a progenitor population that's sort of a wound senescent signature with some inflammatory markers. Pericyte, that's where the vasculature component comes in. And then the cartilage-like one over here is connected with fibrosis. And we can apply this to the clinical data sets now. And we actually do see senescent cells in purple here that are around blood vessels, and there are ones that are more stretched out morphologically, like fibroblasts. And then we can see the genes that drive these interactions. And then I mentioned we looked at cell communication, developing these programs, and how we get communication happening between things like pericytes and macrophages. And where we are now with this is senescent cells and their connection, that we have these pericytes that have a lot of Notch hypoxia markers and WNTs. We have these senescent fibroblasts that are making a lot of collagens and mechanoresponsive elements, right? So that matrix senescent cell is very mechanoresponsive, and they're communicating with various immune cells. So finally, getting to aging, we have these implants or therapies that are engineered in mouse models that are generally young, very inbred, but real life, if we're thinking about clinical trials, we've got aging, sex differences, microbiomes, infection, various comorbidities, and we're looking at all of these things. But I'd like to talk a little bit about the aging environment and diet. So that same material I showed early on that promotes repair, when we put it in older mice, we get a lot of adipose formation and fibrosis happening, and very little regeneration as characterized by central nuclei. So all the nuclei are at the edge of the muscle fibers. So can you still regenerate an aging muscle environment? And I was very inspired by this paper by Ben Cosgrove, who we're partnering with in the SENDNet program. And they took aged stem cells out of the tissue and rejuvenated them in vitro, essentially took them out of that aged environment, put them back in, and you could get good repair. So that gives hope that maybe you can. Now, if we look at just the immune changes with aging, so no injury, no therapy, it's well recognized that, first of all, your lymph nodes, these are the inguinal lymph nodes. Young lymph node is much bigger than the old one. And the contents of these secondary lymphoid structures are different. So it's been well recognized with aging, you lose naive T cells. But it's different in the lymph node, you lose it, but then the spleen, you see even a bigger loss between young as Y and O is old. So the percent of naive cells goes down, and effector phenotypes essentially means that you've become one of those helper T cells making different cytokines. Okay, so the spleen, you've got a lot of these effector phenotypes that means they're making a lot of cytokines. So if we look at the CD4 T cells in the lymph node, you get a lot more interferon gamma and IL-17 in the old. So there's no stimulus here. They're just more inflamed. You have a lot of more cytokine being produced. And then your gamma delta T cells, you get less interferon gamma and more IL-17. Interesting to note, you get more IL-17 with aging. And the other thing we noticed is that the older animals have more gamma delta T cells to start. Okay. What happens with aging? Well, here we have no injury, just a muscle tissue baseline with young in blue and old in sort of an orangey red color. Baseline, you've got a little bit more of your effector cells. Injury increases inflammation. Not a big difference in IL-4, but you'll see in IL-17, you get an increase with just injury itself. And then when we put our biomaterial in there, we get reduced interleukin-4 with those old animals, but it's like a double whammy because we get increased IL-17 in the tissue with that implant. If we look at some other important components, eosinophils help make IL-4. Those are down in the older animals. And we haven't talked too much about CD8 cells, but we have a lot more CD8s in the old animals with injury and with therapy. Now, if we look at the draining lymph nodes, we see changes happening too. And what I think is interesting here is that you might not see too much of a difference at baseline, but the injury uncovers additional immunological abnormalities. So not too much of a difference in the inguinal lymph node with basic aging, but then that injury really blows up that IL-17. Interferon gamma, you've got more of that in both situations. So this is important to that balance I mentioned, where if you have IL-17, it's blocking that regenerative response. We're also curious, is there any difference between an old and a young Th17 cell? So we have various markers that the immune field uses, but what does that mean when you're in an aging environment? So we took young and old spleens, and even though the old animal has fewer naive CD4 T cells, you can still isolate them, right? We can purify them. And then we culture them in standard media or in Th17 differentiation media. So we see more IL-17. Is that just because they can differentiate more or something else? So we see increased effector differentiation in culture, and gamma T is the transcription factor for IL-17. So these old cells are ready to go. But what I thought was the most interesting is that while they both make IL-17 is here, the old cells are making a lot of different things compared to the young, and a lot of things related to angiogenesis, like VEGF. So these T cells from the old animals in the spleen make IL-17, but also other cytokines or other proteins important for angiogenesis. Now we can use our transfer learning technique to look for age senescent cells and compare them to young senescent cells in a single cell data set, which I won't get into. And there's some commonalities, but you get more of the old senescent cells, and what they're making is a little different. So this is that scar-like, cartilage-like senescent fibroblasts, and they make different things. But what we found most interesting is that the T cell senescence communication changes with aging. So in young animals, if we look at the cell communication, we don't see much going on with this pathological senescent fibroblast. A lot going on with endoperiocyte communication. In the old animals, there are not too many signaling connections happening, but those T cells are super active, and these pathological senescent cells are also very active. So it's not just what cells you have there, it's how they're talking to each other. This can help with development of the right therapies because the same thing that works in the young animal might need some extra help in the aged animal. So we put that regenerative material in an old animal, combine it with IL-17 neutralizing antibodies after 1 week. If you block that immune phenotype too early, you reduce migration of immune cells to your injury site, and you do need an immune response. You just need the right one. And if we have that neutralizing antibody, we increase interleukin-4 by T cells and eosinophils. And you can see reduced fibrosis and adipogenesis. So here's the adipogenesis here compared to with the neutralizing antibody. And if you look at gene expression, you can see reduction of fibrosis and adipogenesis markers. And you start seeing centrally located nuclei in the muscle tissue, suggesting regenerative phenotype. So the last story I'll talk about continues with the lessons in translation related to diet. And this is going to bring us back to the gamma delta T cells. So remember, those gamma deltas went up with aging, and they were found in fibrosis. So, another reason we think there's a connection there. So these gamma deltas, again, there's still a lot to learn about what they're recognizing receptor-wise. They have a lot of different functions in modulating immune responses. And like everything in immunology, there are a lot of numbers and letters that are really confusing. V region, gamma regions, and so the V gamma 4 and V gamma 6 are what we're interested in. One being more tissue resident, and the other being more from the blood and migrating into the area. We can pull out the gamma deltas from our single cell analysis or flow cytometry, but essentially the gamma deltas can have regulatory interferon or IL-17 phenotypes, essentially just like T cells. What happens in fibrosis? Well, by flow cytometry, we can compare interferon gamma and IL-17 in our mouse model. And essentially with interferon gamma, your injury and implant goes up and then it comes down, right? That's what happens with interferon gamma. In IL-17, what you see is after that repair process is over, right? So you finish with interferon gamma, you start getting this IL-17 producing gamma delta. This is in a young animal. right? So that IL-17 producing gamma delta starts increasing. And what we see is if we put these animals on a high-fat diet, you significantly increase the percentage of gamma deltas that are in the region. And there's a direct correlation between the weight of the mouse and the percentage of gamma deltas in there. And then the type of gamma delta is different. If we compare the V gamma 4 versus the V gamma 1. And one goes down on high fat and the other one goes up in high fat diet. And interferon goes down, IL-17 goes up, but the type of IL-17 changes. So in the low fat diet, you get T cells that make a combination of IL-17F and A. And in the high fat diet, they tend to shift more to the IL-17A. And this is the more, the tighter binding IL-17. So it's a more active signaling. So what we're thinking about with adaptive immunity, aging, and this tissue composition of how much repair versus fibrosis is happening is in a young T cell responding to this tissue damage, which can be danger signals that stimulate the innate immune system. But we also think there might be some direct recognition of peptides produced during tissue damage, particularly that can be recognized by regulatory T cells that are allowed to recognize self. The young T cells have reduced TORC1, increased TORC2 with the regenerative material. That modulates macrophage phenotype. We have Tregs, type 2 immunity, and you get some productive repair happening. The aged T cell is shifting to a more inflammatory phenotype. You've got interactions with senescent cells making multiple cytokines such as IL-6, reduced TORC1, reduced TORC2, increased TORC1, IL-17, interferon gamma, and more of a fibrosis phenotype. So there are many factors involved in tissue composition. So this is really what we think is going on with at least the immunological aspect of immunological contribution to tissue composition. So one theme that I hope to have gotten across today was this connection between basic research, therapeutic design, and clinical translation. And it's really the clinical translation that impacted our basic research of starting to look at the immune system, which changed our therapeutic design and hopefully clinical translation as we're thinking about factors that influence the immune system and could impact the efficacy of translation. I think we're going to be smarter developing therapies to target those specific factors that can be variable between people, such as diet, comorbidities, aging, sex differences, things that can modulate the immune response. So with that, I'd like to stop and acknowledge the many collaborators and many students, and of course, funding. And I recognize I went through a lot of different topics rather quickly, so I'd be happy to answer any more in-depth questions about any of those topics. So thank you for your attention.
Questions? I'm wondering how the high-fat diet affects the T cell. Do you think it's a direct effect or this signal through the organs? What is that?
Yeah, so both with the aging and high-fat diet, I think inflammation in the gut area is impacting the distal tissue. I'm sure there could be other factors too, but we've got additional studies happening where we put a specific infection in the gut, and we see distal increases in gamma deltas making IL-17 in the fibrosis in the area of tissue damage. We have another study where we're taking young and aged microbiome and transplanting into young germ-free mice. And the old microbiome, we get more gamma deltas making IL-17 in that environment. Now, we have some preliminary data, which hopefully I'll have enough to be able to present soon. In the gamma delta knockout, the fibrosis structure changes. So it was great. So as a student, it can be really frustrating when you don't see differences, right? We did the picoserous and Mason's trichrome, and students like, we don't see a difference. They tried to quantify it, and of course, they're really stressed out, right? Well, we went and sorted CD29 positive stromal cells and did bulk sequencing. And we saw differential regulation of key proteoglycans and collagens. And there were some collagens connected to cartilage. So I was really happy, at least something I knew about, right? And these were collagens involved in the fiber organization of collagen. So you could have the same amount of collagen, but the way it was structured is different. So we're collaborating with Sharon Garecht, who can look at the mechanical properties with some nanoindentation. And we see reduced mechanical properties in the fibrosis in the gamma delta knockout mice. And I think that's really cool. Not just, you know, you think of tissues getting stiffer with aging, but the mechanical properties of the matrix can impact immune cell migration, immune cell activation, right? So CAR T people are looking now at different substrates and culturing CAR T cells ex vivo that are more viscous or stiffer or softer, and it changes the CAR T phenotype even, you know, days after it's been implanted in vivo. So I'm really excited to understand how these infections or different immune cells essentially impact the mechanics of the tissue environment.
So when you're looking at T cells on like old and young mice, what constitutes old and what constitutes young? How do you...
Great question. So we can start seeing differences at 12 months. So that's when you start seeing senescence coming up. But we tend to look at 18 to 20 months. And then, for example, the old microbiome samples, those are from like superagers. And it also depends on the strain of mouse. So, you know, the P16 Cree mice have a lot less P16 and they get a lot more cancers and they die younger. So, you know, the old mice, 18 to 20 months there might be equivalent to like a geriatric mouse and a wild type. So it's a little bit different. And the variability, of course, the heterogeneity is significant there. So usually 18 to 20 months, in part because that's where you can buy them too. Not enough genetics in there, huh? Thank you. So does the implant change its response in an aged animal? And we are looking at that, and there are sex differences. With that, we haven't dug down too much. We've looked at regulatory T cell differences. We haven't looked too much at phenotypic differences, but I'm sure they're there. So yes. Also, if you look at people, I feel like it's a locally aged environment. And in the reporter mouse, we've noticed that when you have more senescence in the tissue, we also see increased senescence in the spleen. And we're quite interested in that. And then the senescence signature that we have in that fibrosis, working with collaborators at NIA who've been looking at signatures of aging in serum, there's a huge overlap. So this is a fibrotic signature in a young animal, correlating pretty strongly with serum proteins, correlating with an aged old mouse that's frail. So I think that's pretty neat. And actually in our clinical implants, you can do a TCR repertoire analysis, and you know it's a real pain, but there are some that are known. We see clones of T cells recognizing CMV, which is also connected, you know, with aging and frailty, and they seem to be expanding around the implant too. So I think there's some interesting intersections, and yeah, an aging tissue is not going to be as intensely fibrotic as those implants, but there seem to be a lot of parallels between the two that I'm really curious about.
So I'm very interested about the structure of this SMEA. Can you just tell us a bit more about the changes with transplants?
Well, we're putting in material, so essentially we're just inducing fibrosis. And actually those are signatures that, again, you see with aging, but even around tumors, and especially tumors that are non-responsive to therapy tend to have that fibrosis. The structure of it changes depending on the condition, and I'm sure there are potential genetic structural differences. So the gamma delta knockout, we had decreased expression of collagen 9 and 11, which are the ones involved in the factogenesis. When we had that microbiome infection with ETBF, we saw significant changes in agarcan, which is a proteoglycan impacting viscoelasticity. So, you have an implant, and the collagen fibers do tend to align around the implant. And it's interesting to think about gamma deltas. One of our collaborators discovered the gamma delta cells, and he discovered them in the context of tuberculosis and the walling off of that caseous thing that forms with tuberculosis. That's where they were discovered. So again, I think not just an infection response, but involved in that fibrosis to wall off. IL-17 is also connected with skin and gut repair. So I think barrier tissues need IL-17 to help maintain the commensal balance and also repair, the skin and the gut, probably a little bit in the lung too. But things like muscle, liver, and brain, like the more type 2 phenotype. So we're just starting with ECM, TEM, transmission electron microscopes of the ECM, where you can actually see the fibers nicely. And so I'm excited about that because, again, our biomaterials background, we actually built materials for the cornea. So you have a lot of collagen, but we would play with how it was aligned so it was clear. But obviously, the collagen in your skin is not clear. The tissue is not clear. The whole structure of the collagen impacts not just the functional properties, the clarity and mechanics, but also the biology.
And can you select changes with accuracy within the tissue that has greater in the position? Is always like greater ECM is greater accuracy or?
So the adiposity seems to be quite interesting, and another example where accidents in the lab can often be good. So a student who had done some work in brown adipose tissue, which is really interesting metabolically. She was using a different method to preserve the samples, so we actually could see the adipose nicely. You actually see some brown adipose at the edge of the injury site in young animals. In old animals, you don't see that as much, and the adipose is just sort of all over the place. Interestingly, in one of our tumor projects, the adipose around the lymph node seems to have interesting immunological changes that I think are important for the lymph node function. And just one more connection, our Hellman collaborator looks at immunological change in adipose tissue with the H. polygyrus infections, that even after you've cleared the infection, those changes persist in the adipose. So, is it a nutrient for the repair process? I think that's an interesting idea, but it seems to be dysfunctional in the aging environment.
And senescent cells have developed ways to evade like immune surveillance, especially like from natural killer cells. Do you know if there's like any mechanism in which T cells would regulate the production of IL-4?
This is really an interesting question because I don't know if I fully agree with that, because when you have an immune response, your senescence goes up. So it's almost like they work together. And it means there's an immune activation, there's something wrong. And that's why you have the immune system in senescence, right? So I think the environment is saying, yeah, there's still a problem, we don't clear us. I'm really fascinated by the CD8, potential CD8 impact. So there was the paper looking at checkpoint blockade, which essentially infected the CD8 cells, and those seem to clear. And we are working with a colleague to look at retrotransposon expression by senescent cells. And how that works is you don't have to have the virus made, you just need some of those proteins that then can be expressed on the MHC complex. And we think those are recognized by CD8 cells. So, and then, those things are involved with wound healing and placental development. So it makes sense that, if you have a chronic wound that isn't healing, that they'll still be upregulated. Now, no one mentioned that, I said Th2 is important for wound healing, but it's also allergy, right? That doesn't make sense. So in our single cell work, we see the Th2 from these materials is different than sort of your allergy Th, or let's say a Th2 population around the CD4 helper cells, and they're close to the Tregs. And we're looking at sort of RNA velocity and trajectory analysis. Are those Tregs becoming Th2s? And they seem to have a regulatory like property. The reason I mention that is we're looking at checkpoint blockade there also. And we see reduced CD8 cells and reduced senescence in those environments. So is the IL-4 Th2 clearing that, or is it just a less inflammatory environment that I did want somebody to do it, but never got done, of like taking the Th17 cells, Th1 cells, and Th2 cells, and Tregs and culturing next to fibroblasts to see that senescence immune interaction. We haven't gotten there. There are tons of NK cells though that are attracted by the biomaterials too. So I didn't give you an answer, but I gave you all the different things that are potentially leading us to an answer.
All right, thank you very much.
Thank you.